Okay, good morning, students. Am I audible?
Good morning, Rajesh, sir.
Good morning, sir. Yes, sir.
Okay. So today we are going to discuss about Krebs cycle. Krebs cycle is also called as citric acid cycle and tricarboxylic acid cycle. Krebs cycle is given two names. It is also called as citric acid cycle and as tricarboxylic acid cycle. Why is called it is called as citric acid cycle and tricarboxylic acid cycle? You will be knowing in the coming slides. So coming to definition, Krebs cycle is discovered by a scientist called as Krebs. It is called as citric acid cycle and also as tricarboxylic acid cycle because citric acid is both the first product and the final reactant in this cycle. And this citric acid contains three carboxyl groups. So because it contains three carboxyl groups, it is called as tri, means three carboxyl groups, tricarboxylic citric acid. So tricarboxylic acid cycle. So because citric acid is the first product and the final product reactant of this entire cycle or cyclical reactions. Here it is called as citric acid because the citric acid is found to be containing three carboxylic acid groups. It is called as tricarboxylic acid cycle. So what does it actually involve? It involves, this Krebs cycle involves oxidation. It is nothing but oxidation. Oxidation of what? Acetyl coenzyme A. So when acetyl coenzyme A is oxidized, it results in the formation of carbon dioxide and water. Okay, we know that carbon dioxide plus water in the presence of glucose give rise, in the presence of sunlight, give rise to glucose and oxygen during what we call it as photosynthesis. And an opposite of photosynthetic reaction is called as respiration. Here what happens? Glucose is oxidized. That means in the presence of oxygen, glucose is broken down to release carbon dioxide and water, during which energy is released. Thus, Krebs cycle is one of the aerobic pathways responsible for the release of energy. And here, before energy is released, it requires another reaction, that is electron transport system. So for that, it produces certain substrates. And what those are, we are going to discuss now. But the overall reaction during Krebs cycle is, it involves the oxidation of acetyl coenzyme A to carbon dioxide and water. So, and how come this acetyl coenzyme A is formed? How is it formed? Let us see. So, it was proposed by Hans Adolf Krebs in 1937 based on the studies of oxygen consumption in Fijian breast muscle. For this outstanding work, he received Nobel Prize in 1953, and also the same cycle, citric acid cycle or Krebs cycle, citric acid cycle or tricarboxylic acid cycle is also given the name Krebs cycle after his name. Coming to the location where this Krebs cycle occurs. The entire Krebs cycle occurs in the mitochondrial matrix because the enzymes responsible for Krebs cycle are present in mitochondrial matrix. Okay. So what actually happens here? We know that aerobic respiration involves three steps. First one is glycolysis, second one is Krebs cycle, third one is electron transport system. In these three reactions, the first reaction is glycolysis. In glycolysis, glucose is converted to pyruvic acid. Glucose is converted into pyruvic acid. And this pyruvic acid does not enter Krebs cycle directly. All these are interconnected. Glycolysis is followed by Krebs cycle is followed by electron transport system. So first glycolysis has to take place. Next it has to be followed by Krebs cycle. And in the glycolysis the end product is said to be pyruvic acid. Pyruvic acid cannot enter as it is the Krebs cycle into the mitochondrial matrix and it cannot participate as in the form of pyruvic acid in Krebs cycle reactions. First of all, it has to be converted to acetyl-CoA. So pyruvic acid that is produced during glycolysis is converted to acetyl-CoA before entering Krebs cycle. That is the metabolic rate of pyruvic acid. So what is the reaction involved here? The reaction involved here is called as oxidative decarboxylation. Oxidative decarboxylation. Excuse me, just a second. So here two reactions take place. One is oxidation, the other is decarboxylation. In decarboxylation, what happens? There is removal of carbon dioxide. So let us see how this takes place. Pyruvic acid, we can also call it simply as pyruvate. Remember, pyruvate or pyruvic acid, both are same. So pyruvic acid or pyruvate produced at the end of glycolysis, it has a number of metabolic fates depending upon the tissue and the metabolic state of that particular tissue. For example, if the tissue undergoes or requires lots and lots of energy, it has to exhibit aerobic respiration. So in aerobic respiration, after glycolysis, what has to happen is pyruvate has to be transported into mitochondrial matrix. And for this, what happens? After the transportation of pyruvate is oxidatively decarboxylated or it undergoes oxidative decarboxylation to produce acetyl coenzyme A. So what is the metabolic fate of pyruvic acid? To produce acetyl coenzyme A. Transport of the first event, that is transport of pyruvate into mitochondrial matrix. How it takes place? Pyruvate enters the mitochondria via a specific pyruvate proton symporter. Okay. So here this is the cell, this is the nucleus, and glycolysis takes place. We all know that glycolysis takes place in cytoplasm. So in this reaction, glucose is converted into pyruvic acid. Now the pyruvic acid has to enter Krebs cycle. The enzymes for Krebs cycle are present in the matrix. Here in the matrix they are present. So pyruvate has to enter the matrix. Pyruvate has to enter the matrix, mitochondrial matrix. How is it possible? From the cytoplasm through the mitochondrial membrane by a specific symporter. Symporter means it is a carrier which transports two molecules or substances from one side to the other side. The molecules to be transported are present on the same side. So here pyruvate and hydrogen ions both are present in the cytoplasm through a carrier protein along with protons. Pyruvic acid also enters the mitochondrial matrix. Then the second reaction in oxidative decarboxylation of pyruvic acid is conversion of this pyruvate to acetyl coenzyme A, and this we call it as oxidative decarboxylation. It is found to be an irreversible reaction. That means once pyruvate is converted into acetyl coenzyme A, it will not be, that means it will not be reconverted back to pyruvic acid. So thus it is one-way reaction. It is an irreversible reaction. And it requires an enzyme. But this enzyme is not a single enzyme. It is a complex. It is a complex. And this is called as pyruvate dehydrogenase. The name of the enzyme is pyruvate dehydrogenase, but it contains some additional coenzymes and cofactors. Hence, we call it as pyruvate dehydrogenase complex. So, thus, this enzyme is a multi-enzyme complex which is present only in the mitochondrial matrix. That is the reason why pyruvic acid, which is actually present in the cytoplasm, cannot be converted in cytoplasm into acetyl-CoA because pyruvate dehydrogenase is present only in mitochondrial matrix. That is the reason why pyruvic acid has to be sent to mitochondrial matrix where this enzyme converts pyruvic acid to acetyl coenzyme A. So coming to the conversion process of pyruvate to acetyl CoA. Here we have already said that, seen that here the pyruvate dehydrogenase complex is a multi-enzyme complex. So it requires 5 cofactors and coenzymes. So let us see what they are. The first one is thiamine pyrophosphate. So what is thiamine pyrophosphate? It is nothing but vitamin B1. Okay, the scientific or the chemical name of vitamin B1 is thiamine. So if you are deficient in vitamin B1 deficiency, okay, what happens? This enzyme reaction will not take place. So you have to take all kinds of food that contains even thiamine. The second one, it requires a lipoic acid. Third one, coenzyme A, because pyruvate has to be converted into acetyl coenzyme A. You also require the substrate coenzyme A. And the fourth one is flavin nucleotide. So flavin is nothing but again vitamin B2, riboflavin, we know that. So it requires two vitamins. We know that vitamins act as coenzymes. And the next one, last one is nicotinamide adenine dinucleotide, NAD, nicotinamide adenine dinucleotide. And nicotinic acid, we know that it is also a vitamin. So thus, it requires 3 vitamins and a coenzyme A and a lipoic acid. These are the 5 factors required for the conversion of pyruvate to acetyl coenzyme A. The overall reaction of pyruvic dehydrogenase is that pyruvate is acted upon by pyruvic dehydrogenase, and during this reaction acetyl coenzyme A is produced, for which the coenzyme A is provided by the multi-enzyme complex, and as the name implies, pyruvic dehydrogenase. Dehydrogenase. What does it mean? D means removal. What does it remove? Hydrogen. So that hydrogens that are removed during this reaction from pyruvic acid are transferred to NAD plus, so that NAD is reduced to NADH plus H plus. As pyruvate is said to be hydrogens, we say that it is oxidized, and during this oxide is removed, hence we say that it is decarboxylation. Thus, by oxidative decarboxylation, pyruvic dehydrogenase converts pyruvate to acetyl coenzyme A, carbon dioxide, and during this reaction, NADH plus H plus. Here, there are two hydrogens, okay, produced. And coming to the overall reaction, just a second, please wait. ఎవరు అసలు కాదు. Okay, sorry for the interruption. So conversion of pyruvate to acetyl CoA. So pyruvate is decarboxylated. That means carbon dioxide is removed to give hydroxyethyl thymine pyrophosphate. Okay, and this thymine pyrophosphate is said to be provided by the multi-enzyme complex, as we have seen earlier. And this reaction is catalyzed by the decarboxylase activity of pyruvic dehydrogenase. So pyruvic dehydrogenase just does not do what we call this reaction, that is dehydrogenation. In addition to the dehydrogenation, it is also having a decarboxylase activity. So the decarboxylase activity decarboxylates pyruvate to give rise to hydroxyethyl thymine pyrophosphate, or simply TPPP. Then the hydroxyethyl thymine pyrophosphate then converts into acetyl lipomide by the activity of another enzyme, dihydrolipoyl transacetylase. We have said that pyruvate dehydrogenase is a multi-enzyme complex. So here there is another enzyme that is required for this complete conversion of pyruvate to acetyl CoA, and this enzyme is dihydrolipoyl transacetylase. So first of all, pyruvate is converted into hydroxyethyl thymine pyrophosphate, and in the presence of dihydrolipoyl transacetylase, hydroxyethyl thymine pyrophosphate is converted into acetyl lipoamide. It is converted into acetyl lipoamide. Okay. So this is the third product that is formed. First one is pyruvate is converted into hydroxyethyl thymine pyrophosphate. This is the first reaction. The second reaction, hydroxyethyl thymine pyrophosphate is said to be converted to acetyl lipoamide. Next, the same enzyme, dihydrolipoyl transacetylase, now catalyzes the transfer of from acetyl lipoamide to coenzyme A to produce acetyl coenzyme A. So in the third reaction, what happens? Coenzyme A comes in, and this acetyl group from acetyl lipoamide is transferred to coenzyme A, so that acetyl CoA is formed. Okay. In this reaction, what is happening? Acetyl is said to be removed from lipoamide. So the reduced lipoamide is now converted to oxidized lipoamide. So it is reduced, and now this is converted to oxidized lipoamide by transferring the reducing equivalents to FAD by an enzyme dihydrolipoyl dehydrogenase, thereby forming FADH2. Okay. So here the FADH2, but actually we have seen that in the overall reaction NADH plus H plus is produced. So what happens is FADH2 in turn transfers the reducing equivalent, that means protons, to NAD to give NADH plus H plus, which can pass through the respiratory chain, that means electron transport system, to give 3 ATPs. Okay. So thus from one it is here. This is the coenzyme A participates, that is thymine pyrophosphate, so that pyruvate is converted in the presence of pyruvate dehydrogenase to produce hydroxyethyl thymine pyrophosphate. And this hydroxyethyl thymine pyrophosphate is converted to acetyl lipoamide in the presence of dihydrolipoyl transacetylase. Okay. And the dihydroacetyl transacetylase transfers the acetyl group to this, so that to the coenzyme A, so that what happens, acetyl coenzyme A is produced. In this reaction lipoamide is reduced and it is oxidized by transferring the hydrogen ions to FAD, so that FAD is converted into FADH2, and in soon FADH2 transfers the hydrogen ions to NAD, so that it is converted into NADH plus H plus, and this enters the respiratory chain, and from each one NADH plus H plus molecule 3 ATP are produced. As 2-pyruvic acid enter the cycle, here from this reaction 6 ATP are produced during aerobic respiration. So far we have seen the conversion of pyruvic acid to acetyl coenzyme A. Okay. Now this acetyl coenzyme A enters Krebs cycle. So the Krebs cycle involves 8 reactions: condensation, isomerization, oxidative decarboxylation 1, oxidative decarboxylation 2, substrate level phosphorylation, oxidation 1, hydration, oxidation 2. So let us see what condensation is. Condensation means it is the reaction, where two different molecules combine, to form another molecule. A plus B give rise to C. That is simply what condensation is. Isomerization. What happens here? One isomer is converted into another isomer. For example, glucose, we know the isomer of glucose is fructose. It is aldose and the another one is ketose. So this conversion of glucose to fructose is called as isomerization. Likewise, here also there is isomerization process involved. And next, third one is oxidative decarboxylation. The same thing, what happens during the conversion of pyruvate to acetyl coenzyme A, the same thing happens in these two reactions. That means here oxidation takes place, that means hydrogen is removed, decarboxylation takes place, that means carbon dioxide is removed. Thus, there are two reactions. Oxidative decarboxylation reactions are 2 in number. And the other one is substrate level phosphorylation. Here, phosphate is added at the level of substrate so that ATP synthesis takes place. And such a synthesis of ATP, production of ATP at the substrate level does not require aerobic respiration. That means it cannot go through the what we call electron transport chain. It need not go through. Here at the substrate level itself, ATP is produced. And another one is oxidation. That means where hydrogens are removed. Hydration, where water is said to be added. Oxidation, the same thing. Another reaction where hydrogens are removed. So let us see one by one. The first reaction is condensation. So Krebs cycle starts with the condensation of acetyl coenzyme A and oxaloacetate to yield citrate. In the first definition, when we defined Krebs cycle, we have said that it, Krebs citric acid is the first product in the Krebs cycle. Hence, we called it as Krebs cycle. Sorry, citric acid cycle. So here, the first thing that is produced here is citric acid. So whenever you come across with pyruvic acid or citric acid, pyruvic acid can be called as pyruvate and citric acid can be called as citrate. So these are the alternate names. So here, whenever you see that there is citrate, you can recognize it as citric acid. So Krebs cycle starts with the condensation of acetyl coenzyme A. So acetyl coenzyme A combines with oxaloacetic acid or oxaloacetate to produce citric acid or citrate. Hence, it was given the name citric acid cycle. And this reaction is catalyzed by one enzyme, and that enzyme is called as citrate synthetase. Citrate synthetase or citrate synthase. Synthase means synthesis. What is synthesis? Formation. So formation of citrate takes place, hence this enzyme is called as citrate synthetase. The second reaction is isomerization. So in the previous reaction, what is formed? Citric acid is formed. So citric acid now enters the second reaction, that is isomerization. That means it is converted into its isomeric form, that is isocitric acid, or it is also called as isocitrate. And the enzyme involved in this reaction is called as aconitase. So in the second reaction, citric acid is converted into isocitric acid. Okay, it is catalyzed by the enzyme called as aconitase. But this is not a single step reaction. It is a two-stage reaction. It requires two steps where citric acid is converted into isocitric acid. In the first reaction is dehydration. That means removal of water. Water is removed. So with the loss of a molecule of water, citrate is first converted into cis-aconitic acid or cis-aconitate. Citrate is converted into cis-aconitate. Okay, and this 6-aconitate is still attached to aconitase. So that now this enzyme, what does this enzyme do? This enzyme now adds a water molecule to cis-aconitate. So that cis-aconitate is converted into isocitrate. So this is nothing but just an internal arrangement of the hydroxyl groups. For example, if hydroxyl group is present here, okay, in this isomer, in this substance, in its isomeric form, it is arranged, internally rearranged so that it is transferred to another carbon. So that here citrate now becomes isocitric acid. So in the first reaction, citric acid is formed. In the second reaction, it is converted into isocitric acid. And this is called as isomerization. This enzyme involved in this process is called as aconitase. And the third one is an oxidative decarboxylation. We have seen that there are two reactions here, and that is the reason why we call it as oxidative decarboxylation 1. So what happens here? Isocitric acid is oxidatively decarboxylated to produce alpha-ketoglutarate acid. So first of all, what happened? Acetyl coenzyme A combined with oxaloacetic acid to produce citric acid. Citric acid is converted into its isomeric form isocitric acid. Now isocitric acid is converted into alpha ketoglutarate. Okay, and this reaction is an oxidative decarboxylation, and this is catalyzed by an enzyme ortho isocitrate dehydrogenase, because the substrate is isocitric acid and it is a dehydrogenase enzyme. Hence, it is called as isocitrate dehydrogenase. So, isocitrate dehydrogenase converts isocitric acid to alpha ketoglutarate. And this is again a two-stage reaction. Like the previous isomerization, it is also a two-stage reaction. The first reaction is oxidation. The second reaction is decarboxylation. In first reaction, that is oxidation, what happens? Isocitrate dehydrogenase, the same enzyme, isocitrate dehydrogenase, oxidizes isocitrate to beta keto acid intermediate, which is called as oxalosuccinic acid. Okay, the first product that is produced due to oxidation is oxalosuccinic acid. So we are saying that it is oxidation. That means hydrogens are removed, and these hydrogens are accepted by NAD. So that NAD is converted into NADH plus H plus. So here what happens? Oxidation has taken place and NAD is reduced. The second one is decarboxylation. That means remove oxide. So here the oxalosuccinic acid is decarboxylated. That means carbon dioxide is removed from oxalosuccinic acid so that alpha ketoglutarate is formed. Okay, and it is the first step where carbon dioxide is generated. So far we have seen that oxaloacetic acid and coenzyme A, acetyl coenzyme A combine to form citric acid or citrate. Citric acid isomerizes to form isocitric acid. Isocitric acid is converted first to oxalosuccinic acid and then finally to alpha ketoglutaric acid or simply alpha ketoglutarate. And this reaction, and now alpha ketoglutarate now undergoes another oxidative decarboxylation. The same thing happens here. That means here also hydrogen is removed and then NAD accepts it and carbon dioxide is removed. Carbon dioxide is also removed. So how it takes place? Let us see. In another oxidative decarboxylation reaction, alpha ketoglutarate is converted into succinyl coenzyme A. Okay, so alpha ketoglutarate is converted into succinyl coenzyme A, and this is catalyzed by a multi-enzyme complex. And this enzyme is given the name alpha ketoglutarate dehydrogenase. Remember, wherever there is an oxidation reaction, the enzyme is always dehydrogenase. Because it removes mode, we say that it is oxidized. When hydrogen is added, we say that it is reduced. Here, from this, hydrogen is removed, hence it is called as oxidation reaction. And this requires an enzyme called as alpha ketoglutarate dehydrogenase. So, as in the case of pyruvate dehydrogenase, it also requires thymine pyrophosphate, lipoamide, NAD, FAD, and coenzyme A. So, whatever the reactions we have explained during the conversion of pyruvate to acetyl coenzyme A, those reactions do take place again here in the oxidative decarboxylation of alpha ketoglutarate, so that here what happens, two NADH plus H plus molecules are produced and two carbon dioxide molecules are said to be removed. Hence we call this as oxidative decarboxylation 2, and the end product of this reaction is succinyl coenzyme A. Now what happens? Now we have succinyl coenzyme A, and the next reaction is substrate level phosphorylation. Here what happens? Succinyl coenzyme A is converted to simply succinic acid or succinate, and the enzyme involved here is succinate thiokinase. So succinyl coenzyme A is converted into succinic acid or succinate. It is catalyzed by succinate thiokinase. And this reaction, during this reaction, what happens? It is coupled with the phosphorylation of GDP to GTP. So in this reaction, what happens? Guanosine diphosphate, okay, is added with an inorganic phosphate so that it is converted into guanosine triphosphate. And this guanosine triphosphate now removes its or releases its phosphate, and this phosphate is added to ADP so that ATP is produced. So addition of phosphate to ADP is called as phosphorylation. So that ATP is converted into ADP. ADP is converted into ATP. Adenosine diphosphate is converted into adenosine triphosphate. So this reaction takes place at the substrate level. Hence this is called as substrate level phosphorylation. Okay. So during this reaction what happens? Succinyl coenzyme A is converted into succinic acid. So if these are two molecules, here two succinic acid molecules are produced and two GTP are produced, which are converted into two ATP molecules. Conversion of GTP to ATP is said to be catalyzed by a nucleoside diphosphate kinase. And the next reaction is oxidation. So here there are two oxidation reactions. This is the first oxidation reaction. So now we have seen the conversion of succinyl coenzyme A to succinic acid, and we have succinic acid now. So the succinic acid or succinate is oxidized. That means here also what happens? Oxidation means hydrogen is said to be removed. So succinic acid is oxidized by succinate dehydrogenase. So dehydrogenase is the reaction where D means removal, hydrogen is removed here. When hydrogen is removed, we say that it is an oxidation reaction. So succinate is oxidized by succinate dehydrogenase to produce fumaric acid. Fumaric acid or simply fumarate. Okay. And during this reaction, what is happening? Here there is production of FADH2. So flavin adenine dinucleotide combines with the hydrogen that is removed to produce FADH2. Next reaction is hydration. Dehydration means removal of water. Hydration means addition of water. So hydration, here addition of water takes place. The enzyme involved here is fumarase. Fumarase catalyzes the conversion of fumaric acid to malic acid. So here what happens? The fumaric acid that is produced here, the fumarate that is produced here is converted into malic acid or malate. How it is produced? By the addition of one water molecule. Hence we call this reaction as hydration. And the next reaction is oxidation too. Here malate is oxidized. That means here also hydrogen is removed. That means the enzyme here is involved is dehydrogenase. So dehydrogenase acts upon malate. So here this enzyme is called as malate dehydrogenase. So malate dehydrogenase oxidizes malate to form oxaloacetic acid. So in the first reaction we have seen that acetyl coenzyme A condenses with oxaloacetic acid to produce citric acid. So we call this as a cycle. Krebs cycle we call it as. What do you mean by cycle? Cycle means it comes to the same point. It starts here and comes to the same point. So it started with oxaloacetic acid and now it ended with the regeneration of oxaloacetic acid. So oxaloacetic acid is said to be formed. During this, the hydrogen that is removed is accepted by NAD and NADH plus H plus is said to be produced. And this oxaloacetic acid now combines with the second acetyl coenzyme A, so that citric acid is said to be formed. And we have defined in the first thing that citric acid is the first product and the final reactant. That is the reason why we name this as the citric acid. So in this entire reaction, what happens? Coming to the summary of these reactions, acetyl coenzyme A is converted ultimately to release coenzyme A, and during this reaction, two carbon dioxide molecules are removed. And during what happens here, GDP is undergoing substrate level phosphorylation to produce GTP. Later, GTP is converted into ATP. At the same time, there are dehydrogenase activity during which hydrogen is removed, and this hydrogen is accepted by FAD and NAD. So what happens here? NAD participate and FAD participate, and 3 NADH are produced and 1 FADH2 is produced. So in this entire reaction, can be summarized as this: acetyl coenzyme A plus 3 NAD plus 1 FAD, 1 GDP plus 1 phosphate molecule give rise to 3 NADH. That means NAD is reduced. 1 FADH2. FAD is reduced. GDP and inorganic phosphate combine to form GTP, substrate level phosphorylation. And during this reaction, carbon dioxide is removed and coenzyme A is also removed. So all these are said to be recycled. So the carbon dioxide is said to be sent out through the lungs, respiration. Okay, and GTP is converted into ATP, and coenzyme A that is produced here e-recycle back. So the pyruvic acid is converted again to acetyl coenzyme A, so that the Krebs cycle goes on. And then what about the NADH and FADH2? For the second cycle to go on, we require NAD and FAD, but not NADH and FADH2. So what they have to do? They have to be converted to NAD, so that hydrogen is removed. FAD has to be converted to FADH2 has to be converted to FAD, so that hydrogen is removed. So these hydrogens that are removed during these reactions, what we say, when hydrogen is removed, we say oxidation. When hydrogen is added, it is reduction. So here NAD is reduced, FAD is reduced, and now these have to undergo oxidation. That means removal of hydrogen, so that they can come back to their normal position, normal level, so that they can participate again in Krebs cycle. And this reaction where NAD and FAD are oxidized takes place in the respiratory chain named as electron transport system, which we are going to discuss tomorrow. Coming to the products of citric acid cycle. So what are the end products of citric acid cycle? The end products of citric acid cycle are, these are recycled, this is removed, and 1 ATP is formed. So but what is that is required for electron transport system? These two. So the NADH and FADH2 are the vital products of the citric acid cycle. They have to undergo oxidation. And who is going to oxidize them? It is the oxygen. It is the oxygen that is responsible for the reoxidation. And this is accomplished during what we call the production of ATP. How ATP is produced? Where ADP is added with one inorganic phosphate, it is converted to ATP. And this we call it as phosphorylation. So there is oxidation of these two followed by phosphorylation of ADP to produce ATP during electron transport chain. We call this as oxidative phosphorylation. Okay, and Krebs cycle is called as an amphibolic pathway. It is called as amphibolic pathway. Why is it so? The citric acid cycle is catabolic because it involves degradation, isn't it? It involves degradation and is a major free energy conservation system in most organisms. So here, citric acid is broken down to produce a number of products, isn't it? Hence, it is a catabolic process. And during this, carbon dioxide is released and hydrogen ions are removed. At the same time, several biosynthetic pathways utilize the intermediate products of citric acid cycle. So we have seen citric acid is formed, it is converted into isocitric acid, it is converted to alpha ketoglutaric acid, which is converted to succinyl coenzyme A, that is converted to succinic acid, then fumaric acid, then malic acid, finally oxaloacetic acid. So these are the different products that are produced during the entire cycle. And these products are utilized for the synthesis of other substances in another metabolic activity. So synthesis is anabolism degradation is catabolism. When both occur, because they both occur in Krebs cycle, this is called as amphi means both, amphibolic pathway, because both anabolic reactions occur here and catabolic reactions also occur here. So this is the overall cycle which you have to remember. What happens here? Acetyl coenzyme A undergoes condensation in the presence of citrate synthase, okay, with oxaloacetic acid. That means these two undergo condensation in the presence of citrate synthase to produce citrate or citric acid. Citric acid is converted into its isomeric form. Isomerization takes place. So that what happens? Cis-aconitase is first formed, followed by the formation of isocitric acid. It is a two-step reaction. Both are catalyzed by aconitase only. And now the third reaction is a oxidative decarboxylation reaction. Here, sorry, third reaction is a dehydrogenation reaction. Here what happens? Isocitrate dehydrogenase acts upon isocitric acid so that it is converted into oxalosuccinic acid. During this, the hydrogens that are removed by dehydrogenase are accepted by NAD so that NADH plus H plus is produced. And now oxalosuccinic acid is there. And this oxalosuccinic acid now undergoes, here oxidation has taken place. Now what happens? Decarboxylation, removal of carbon dioxide takes place. So the same enzyme is responsible for oxidation as well as decarboxylation. Hence we call it as oxidative decarboxylation reaction 1. And during this reaction, one carbon dioxide is removed and alpha ketoglutaric acid is formed. And now what happens? The same alpha ketoglutaric acid is now again undergoing oxidative decarboxylation 2. Here carbon dioxide is removed. At the same time, hydrogen is removed, which is accepted by NAD so that NADH plus H plus is produced. So in these reactions, here NADH plus H plus is produced here during the conversion of isocitric acid to alpha ketoglutaric acid, and during the conversion of phosphoketoglutaric acid also NAD is produced. And during this, what happens? Coenzyme A is added so that alpha ketoglutaric acid, after the removal of carbon dioxide and hydrogen by the alpha ketoglutarate dehydrogenase complex, is converted into succinyl coenzyme A. And the succinyl coenzyme A now undergoes what we call it as substrate level phosphorylation in the presence of succinate thiokinase, where coenzyme A is removed. Coenzyme A is removed and it is converted into succinic acid. During this reaction, GTP is first formed, followed by kinase activity, nucleoside phosphate kinase activity, which is converted into ATP. And the succinic acid now undergoes dehydrogenation again, that means oxidation 1. Here in the presence of succinate dehydrogenase, it is converted into fumaric acid. And now fumaric acid is said to be undergoing dehydration reaction, that means water is removed and it is converted into malic acid. And malic acid is catalyzed by malic dehydrogenase, that means here also hydrogens are removed and this is accepted by NAD so that NADH plus H plus is produced, and this is in this reaction oxaloacetic acid is regenerated. So in this entire process, oxaloacetic acid enters and oxaloacetic acid is regenerated, and this regenerated oxaloacetic acid takes part in the condensation reaction with acetyl coenzyme A to produce citric acid. The citric acid is the first product and the final reactant in this entire reaction. Hence, it is called as citric acid cycle. And all the citric acids and other acids that are produced are having 3 carboxylic acids in their structure. You see the structure, wherever we come across with, there are 3 carboxylic groups, isn't it? Hence, this cycle is called as tricarboxylic acid cycle. Because it is discovered by Hans Krebs, it is called as Krebs cycle. Coming to the energy budget of Krebs cycle. So, it is dependent on P is to O ratio. The P2O ratio refers to the number of inorganic phosphate molecules utilized for ATP generation for every one atom of oxygen consumed. That means how many oxygen molecules are utilized and how many inorganic phosphates are utilized to produce ATP is what P2O ratio is. More appropriately, P2O ratio represents the number of molecules of ATP synthesized per pair of electrons carried through electron transport chain. So here, pair of electrons, why we are saying hydrogen? We know that it is having said to be electrons, two electrons. And for each pair of electrons that are transported, one ATP molecule is said to be produced. And P2O ratio of 3 is assigned to NADH. That means when NADH releases its hydrogen, which gets oxidized, that means which combines with oxygen ultimately to produce water, and during which ATP is produced, the number of molecules of ATP that are produced during reoxidation of NADH is said to be 3. That means the P2O ratio for NADH is 3, whereas the number of ATP molecules produced during the reoxidation of KDH2 is said to be 2 ATP. So the P2O ratio is 2. Thus, from NADH, when it enters the electron transport chain, from each molecule 3 ATP are produced. That means if 10 NAD are present, NADH plus H plus are present, then we get 30 ATP. If only 5 are present, 5 into 3, 15 ATP will get. Likewise, when FADH2 enters the electron transport chain, it produces 2 ATP. That means if 2 FADH2 are produced, 2 into 2, 4 ATP are produced. So that is what the energy budget is. So let us see uh, how many what we call ATP can be generated when the reduced FAD and NAD enter electron transport chain. So during the process of oxidation of acetyl-CoA by citric acid cycle, how many NAD are produced? 3 NADH plus H plus are produced and 1 FADH2 is produced. That means 3 into 3. From each NADH plus H plus 3 ATP are produced. That means 3 into 3, 9 ATP are produced. Isn't it? From 1 FADH2, 2 ATP are produced plus 2. In addition to this, there is one substrate level phosphorylation where GTP is produced which is converted into ATP. That is 1 ATP. Thus, 1 plus 9 plus 2. We come across with the production of a total of 12 ATP produced from one acetyl coenzyme A. From glycolysis, during glycolysis, glucose produces 2 pyruvic acid molecules. So when these 2 pyruvic acid molecules are converted into 2 acetyl coenzyme A, what happens? 2 into 12, 24 ATP are produced. 24 ATP are produced. So thus, coming to the energy yielding Krebs cycle, oxidative decarboxylation of pyruvic acid. That means the first reaction before it enters the Krebs cycle. There 3 ATP and 3 NAD are produced. One NAD is produced, so that 3 ATP are produced. And in the overall reaction, 12 ATP are produced. Thus, it generates, after glycolysis, oxidative decarboxylation, that means conversion of pyruvate to acetyl-CoA produces 3 ATP when it enters the electron transport chain. And the Krebs cycle produces 3 NADH, 1, 2, and 3, 1 FADH2. When they enter, they produce 11 ATP and one substrate level phosphorylation, 1 GTP, produces 1 ATP. Thus, total 15 ATP are said to be produced per molecule of acetyl coenzyme A. So, if there are 2 acetyl coenzyme molecules, the total number of ATP that are produced in Krebs cycle will be 30 ATP. Okay, so this is all about Krebs cycle. Hope you understood well. If any doubts, please ask me. Otherwise, we'll close here. Emma, విన్నారా అందరూను?
Yes, sir.